My talk is a fully Bayesian world, and for some reason I was writing that title down. I couldn't get a song out of my head. What a wonderful world it is. Louis Armstrong. So I'm going to talk about Bayes, and it may surprise you what I have to say about this question. We'll see. The question is, what is your prior probability on what I'm going to say? What is a fully Bayesian? I think at the end, let me go to the end, I'm not so sure we would all agree on what it means to be fully Bayesian. I'm sure we would disagree on that, so I'm going to lay out my ground rules for what I think it means to be fully Bayesian. A complete specification and explicit writing down of priors, utilities, likelihood functions, models, carrying through the analysis of a posterior distribution, calculating expected utilities, optimal decisions, using this to carry through making decisions. And I don't mean by this, everybody here is a Bayesian in the sense that you made a decision to come here today, you looked at the losses, the other things you could have done, you didn't write it down explicitly. So I'm going to ignore the fact that everybody's a Bayesian. Some of us write it down and go through and are fully Bayesian. So that's how I'm going to carry out from that perspective. Inherently, it's subjective. When we go down here and we write down a prior, even the specification of the models, this is going to be a single entity representing the views of this. And now you can imagine how this gets complicated when you're trying to look at the public policy view of this. But it's very much a subjective approach. There may be multiple single agents within a decision process, and we may look at that as well. And of course, we can disagree on what that means. So who is drug development and regulatory? That was part of the question. So when I look at this, there are multiple facets of this. There are drug and device companies, private sponsors, for-profit sponsors trying to develop drugs or devices. I'll talk about both, and the landscape for each one of those is different. And I'll talk about academic centers, certainly run and sponsor clinical trials, carry out clinical trials, investigator-run clinical trials. They're a big part of this. Technology, and I probably would use a worse word for this other than the fact that I fall into this group, meaning CROs, consultants, investors. Investors play a role in this. I'm a consultant for Berry Consultants. I do consulting for both of the top groups, for multiple groups, but involved in drug and device development. Review, and this is where I think much of the controversy is going to come. What do journal editors who play a role in this picture for sure? What gets published, what they allow in journals. Grant review and funding of trials. Regulatory plays a big part of it. I'm from the US, and much of my experience is US dominated, so I'll talk more about the FDA. Which allows me to skirt Rob a little bit. And payers. Payers play a critical role in this as well. So the idea of drug development and regulatory is a big scope here. So drug companies. So Karen's going to talk about drug companies. She's in a drug company, and so she can talk about this much more than I. But from my experience with working them, what role do they play, and do they make decisions from a Bayesian way? Now, we have this old picture of people sitting around a room and just making a decision. Do we make the go, no-go decision? I think that's changing. I know that's changing. We now have explicit utilities written down for moving forward. We have explicit prediction of a phase 3 trial. If I run this trial, what's likely to happen? What is the value of each of those outcomes? What is the utility of those? And of course, it's a monetary decision on their part for sure. It's a complex set of decision problems. The whole program is now being simulated. When you start from the very, very beginning going into phase 1, they look at this whole sequence, phase 1, phase 2, phase 3. They look at the sequence of trials they could run. They simulate that entire process. They may even ask the question, you know, if I did this trial before that one, that's a critical thing I need to know, and I may save money and time in the whole process. Simulation of the process, understanding the trial within the whole scope of drug development. This is done across portfolios of drugs. This is being done explicitly and at a very high level within companies, and Karen can talk more about that. Eli Lilly certainly does a lot of this. It is being done. I don't think it's done enough. I think there's room here to be more Bayesian. There are no roadblocks to this. There's no reason companies can't do this, carry through a decision process. Nobody's saying, oh, you can't do it this way. I actually think many of the roadblocks are training and statisticians. This may be a recurring theme to this, that many times statisticians get in the way of doing this. Why aren't we leading this type of decision problem? We do this better than anybody. We should be at the forefront of doing it, and we're not, in many cases. Trial design. Now, that's the whole decision process, go, no-go decisions, 1 to 2, 2 to 3. All of these processes are internal decisions. They have to be done in a Bayesian way. What about trial design? How many times do you see trials where the final analysis is a posterior probability, is a utility calculation? It is being done. It is absolutely being done. Here's a Lilly diabetes phase 2-3 design. 3 papers are coming out on this in November. It is a trial that starts in a phase 2 stage, has 7 doses, 2 comparators, 1 active, 1 placebo. During phase 2, it's tracking 4 endpoints. This is prospectively defined. The 4 endpoints are combined in a utility function. Company laid this out, spent a lot of time developing a utility function for the 4 endpoints. HbA1c change, weight loss, blood pressure, and heart rate changes. Historical data was used to use longitudinal modeling of early endpoints to longer term endpoints. In a phase 2 trial, they were driven by what happens at 12 months, but they needed to know early how it related to that, so they used historical data and subjective priors on some of those relationships. The decision to advance from phase 2 to phase 3 was based on the expected utility of each of the doses. The posterior probability doses were better than other doses, and the predictive probability, Bayesian predictive probability that the doses moving forward would win the phase 3 component of the design. If those conditions were met, it moves into phase 3 seamlessly, from Monday to Tuesday, perhaps. There were multiple decision makers, and the prediction recognized that Lilly wasn't the final decision maker, that FDA and EMA were the final decision maker and knew the analysis that they were going to do at the end, so it was predicting a different decision maker who has their own criteria and how they make a decision, which, by the way, was a p-value. The trial design was simulated under hundreds of scenarios. What-ifs were played out. Prior distributions were created from the PK/PD group on what they expected the dose to be for each of the circumstance. Variability was built into the parameters. We simulated many sample size, many rules for moving forward. The value of the design was measured by the likelihood of all the outcomes moving to stage 2, not moving to stage 2, depending on what the truth was. This was all done. This was developed. The trial is currently running. The results, I believe, will come out soon. I believe this is as fully Bayesian as I can see a trial being. It was accepted by the EMA. It was accepted by the FDA, and I put an asterisk near that they differed on how they were going to analyze the final data. I think the EMA was slightly more Bayesian in that sense, but there were differences here, but the trial took into account both of them, and it knew how each was going to make their decisions. So my point to that is there were no hurdles there. It was a lot of work. We went to both agencies, discussed with both of them. It can be done. It can be done. Academic centers, I think they're likely far less Bayesian, which in a way is surprising. I think there are different things going on within academic centers. I don't know if it's fear of funding decisions, doing things differently. Strangely, I think that pharma is leading the Bayesian push forward rather than the academic centers. What are the hurdles? I think the question is, are there enough statisticians trained to carry out a Bayesian trial? Is it the statisticians that are the roadblock? I'll talk more about that. Two relevant projects within academia, the AdaptIt project and I Spy 2. The AdaptIt project is a grant funded by the FDA and the NIH, a joint funded project, to the University of Michigan, the Medical University of South Carolina, LA Biomed in Los Angeles, and Berry Consultants. The idea is to bring Bayesian and adaptive techniques to 5 possibly funded, federally funded trials as part of the Neurological Emergency Treatment Trials Network. These are trials that will be submitted for funding. So we go into the 5 trials and we bring Bayesian adaptive techniques to the 5 trials, see how much havoc we can wreak to that. The 5 trials, one is status epileptic, stroke, so this is the treatment in the emergency room of epileptic seizures that are not helped by the first round of drugs. 2 different stroke trials, a trial for cardiac arrest, individuals that come to the emergency room, they've gone through ROS. Spinal cord injury are the 5 trials. The trials have multiple adaptations, modeling of early endpoints, utility functions around different endpoints, modeling MRS in neurology trials. There's a big question is how do we do this? So we built utility functions on the various states of the modified Rankin score rather than dichotomizing it, things like that. We use a great deal of prediction during the trial. Enrichment, it's looking at subgroups, possibly dropping subgroups. And these are all confirmatory trials. Part of this trial is to study the barriers. NIH wants to know why aren't more adaptive Bayesian trials being done. FDA would like more of this being done, so they are studying. They have methodology people watching all of the interactions, taking notes, carrying through, doing surveys throughout the course of all of this on what people think and trying to measure the barriers to adaptive and Bayesian trials. Frequently, perceptions here is typically, nobody stands up and say, I don't accept this. Somebody above me won't accept it. Journal editors won't accept this. FDA won't accept this. NIH won't fund this. It always, and NIH says, well, we just don't get it submitted to us. So it's interesting, this whole part, which is a fascinating part of this project. The I Spy 2 trial is, many of you probably know of this trial. It is funded by the Foundation for the NIH, FDA, and a biomarkers consortium, as well as private sponsors. It is a clinical process. To call it a trial is probably a misnomer. It's for women with newly diagnosed locally advanced breast cancer. And it's a competitive phase 2 process. And what I mean by that is a trial tends to be a finite period of time, and this is a process that may go on forever. It may be the future of clinical trials. There may not be trials, there may be processes. Different therapies move into the trial at different times. They're adaptively randomized within 8 subgroups of tumors, depending on which of the treatments are doing better in that group. The primary endpoint is a Bayesian predictive probability that phase 3 will be successful. Thank you. That phase 3 will be successful. It's built on multiple imputation from earlier markers to longer term markers using historical data in that piece. It's run completely by Bayesian machine, not a p-value within the whole thing in doing that. And it's really a revolutionary idea. What about the lurkers? I'm running out of time, so I'll skip to probably the end of this. Berry Consultants is a company of 9 statisticians now. And that's 9 statisticians. We're growing rapidly. We're essentially 100% Bayesian. We only do Bayesian projects, and we can't keep up with the demand. Things going on in all kinds of directions. And so when I see the question, why isn't this a Bayesian world? It is. It is. All phases, we've worked in all phases. We've worked in over 50 different indications, therapeutic areas. We have software that's entirely Bayesian driven. Underlying part of it is all Markov chain Monte Carlo, all of the things that you can do in simulating clinical trials, which is licensed to 6 pharma companies, 3 CROs, entirely Bayesian driven. Review, I'll skip review agencies and jump to regulatory. Regulatory is interesting, and not just because Rob's here, but I'll defend them from the standpoint that they get a bad rap. I think people, FDA, EMA doesn't accept this stuff. And in part because you don't see a lot of it. But I don't think they're saying no to it. I don't think they're getting it for review. We go with many Bayesian designs and we're rarely told no. We've been told no. We've been told no Bayes here. But it's very, very rare. And yet I hear people all the time saying, oh, FDA doesn't like this. And I hear the exact opposite from them. They have a guidance document on the use of Bayesian Statistics, the Center for Devices and Biologics. EMA has a guidance document out on adaptive designs, guidance document from CBER and CEDAR of the FDA for adaptive clinical trials, and they have a section in there on Bayesian statistics and the use of Bayesian statistics. Phase 1, phase 2, phase 4, completely available for fully Bayes. Up to the users what they'd like to do. I see no hurdles in these types of trials. And actually, in many cases, the FDA is asking for these things. Better phase 2 trials, better decision of the go, no-go decision. It's part of the critical path initiative. They're asking you to do these things. There's some heterogeneity in who you run into in the various teams. You may run into a stumbling block now and again, but it's fairly rare. The difficulty, of course, is the phase 3 confirmatory trial. Where does Bayes play a role there? It's very rare outside of CDRH, the Center for Devices. It's actually quite common to have a pivotal, adequate and well-controlled Bayesian trial, posterior probabilities of the final analysis within CDRH. I think there's reasons here that get the Bayesian upset at this. Let me try to say this before I trip on this. Simple Bayesian trial, you might construct and you go with the FDA single arm trial. If pi is greater than 0.5, it's an approvable thing. That's discussed, the utilities are set. Posterior probability cutoff of 0.95 given 100 patients. That's a successful trial, it's agreed upon, that may be a way that you go. Means 59 or more patients, you can back out. It never was an input to this, but you can back out the type 1 error. It may never be, it's never an input to the Bayesian, but it's an output. Now, 2 things that could happen. Of course, I may say, okay, a posterior probability of 0.95. If I get that same threshold at 50 and 75, that should be approvable. But if I calculate the type 1 error of that design, I of course inflate type 1 error, which was never a part of the Bayesian design. And that's a struggle for agencies. So I might adjust it from a Bayesian perspective to change the threshold to meet a type 1 error constraint. It violates the likelihood principle. 2 trials could be run, exactly the same data from start to finish. 1 trial is a success, 1 trial is a failure. It violates the likelihood principle, which is a theorem to a Bayesian. This is problematic, and the reason is we have to meet frequentist type 1 error constraints when we go to the FDA. It's a type 1 error. It's a type 1 world. Same thing happens with priors. You try to use priors, inflate type 1 error. These are the 2 areas where this issue comes up, and the issue is type 1 error constraints. If you have to deal with type 1 error constraints, it's a frequentist criteria. There's no Bayesian resolution to it. And so all I ask is that each problem possibly be different. Orphan indications are different than something that's going to be used in millions of people. And that's typically not done. Usually it's 0.05, and that's the constraint. So my conclusion, as I'm being swept off here, is that usually the Fullian approach is harder, and it may be the simple Bayesian optimal solution is to do an easy trial. I didn't write it down explicitly, but let's just do an easy trial. Otherwise, it's harder, it's more work, but there are far less hurdles than I think people perceive them to be, and I see trees of green and red roses too. It really is a Bayesian wonderful world if you want it to be. So I'll quit now.
So personal views, primarily personal views. I'm not going to hide behind anything else, but with a sprinkling of sort of general regulatory thinking and enough negativity to make sure we have a debate. Actually, there's a huge parallel between what Scott's saying and what I'm saying, but let's fall down on different sides, otherwise it'll be dull. 4 points of contact with regulators. I'm going to speak about European regulation, obviously. We write guidelines, therapeutic area and methodological, and that's really to try and set some basic standards for what we expect to see in terms of drug development, as I say, from a methodological perspective or in terms of a particular therapeutic area, both to agree them with sponsors and people conducting trials, but just as importantly to agree them amongst ourselves. We live in a complicated, heterogeneous world, which I think was your world, Scott, particularly in Europe, where we're bringing together expertise from 27 different member states, 28 from next year. So writing guidelines is as much to agree the standards amongst ourselves as to communicate with the outside world. Scientific advice is when a sponsor company has a set of questions about a particular development program and they want to discuss those questions with the regulators to get something formal down in writing to say we agree with what you're doing or we don't agree, please consider the following or whatever. So it's a dialogue between sponsors and regulators about a specific development program. Marketing authorization application is when you've actually completed your set of trials, you've written them all up, you've got tens and tens of thousands of pages of information, and you send it into the regulatory agency and you get a yes or no answer at the end. A bit more complicated than that. And then informal interaction, of course, at conferences and scientific meetings. And where do the regulators get involved with drug development? So, clinical drug development. First in man trials, there's a clinical trial approval system for the clinical trial to be conducted in your area, so that's a national competence. So we look after that for the UK, obviously. Exploratory development, the main interaction tends to be around scientific advice, and the type of question from the company is, do you like our direction of travel? Are you happy with the way that we're selecting the dose? Then there's a go or no-go decision, where there's no real interaction with regulators at all, entirely a sponsor decision. Confirmatory trials, which are often the main subject of scientific advice, and then of course the main subject of a marketing authorization application and the assessment, and then post-marketing and life cycle work, which obviously also comes for review and for updating the product license as required. To touch on the first of those 4 points of interaction, guidelines, not an awful lot about Bayesian methods in regulatory guidelines. In ICH E9, the International Conference on Harmonization E9, which is a global guidance document, this is the limit of what you can find. So there's a definition of Bayesian statistics at the top, which I guess is fairly familiar to us all, and an excuse at the bottom. And the excuse is the guidance largely refers to the use of frequentist methods because that's what everybody does. I don't know that it's particularly welcoming or optimistic about your chances of success if you use a Bayesian method. It doesn't infuse. As I say, it's maybe a bit of an excuse. Other approaches may be considered when the reasons for their use are clear. It looks like you've got to have a special reason not to use the frequentist model. So I can understand that that's not particularly welcoming. What do we see in practice? Very much echoing Scott's thoughts that the answer is basically not a lot. The dialogue that we have about exploratory development, and I've put modelling, and that's in the context of dose finding, dose response, in exploratory development is where we see the most use of Bayesian techniques. No problem at all. And I can take 20 seconds to clarify one of the many misconceptions about regulators. You'll often hear people saying that the regulators don't care about dose response. It's the company's risk. Of course we care. The point is, if the company does something silly, we can't do anything about it, and that's why it's the company's risk. It doesn't mean we don't care. But okay, modelling and exploratory development, anything goes, no problem. We have got a handful of examples of using, let's call it a fully Bayesian approach in confirmatory development, but it really is a handful, literally. I don't think we've endorsed any proposal to use a Bayesian method, and I mean here in the context of using an informative prior for the estimate of the treatment effect, for a primary analysis of a confirmatory trial. And that's, if you like, that's almost the pinnacle of the information that the regulators are going to use for their decision. I don't think we've endorsed that. But within the context of confirmatory trials, again, echoing what's already been said, we certainly have endorsed the use of Bayesian models for decision-making analysis, and the Lilly diabetes trial is one example of that. The rest of the text on the slide relates to the context in which we've seen the approaches, and I perhaps won't say too much about this now. Hopefully the thinking becomes clear in later trials. A company might come and say, look, this is a difficult situation, we can't recruit the number of patients we want to be able to get in order to perform a frequentist analysis with a sample hypothesis test and a sample size. So therefore we want to use a Bayesian method, get a chunk of information from an informative prior and just sort of top that up with whatever we can find. And that might be a genuine problem, or it might be a not so genuine problem as an excuse to go and collect less information. I might come back to that. And then the third point I put up about marketing authorization applications. This is the licensing step, this is the decision point. Here, for those who aren't familiar with the criteria you use to make a licensing decision, these are the only reasons that we can reject an application. Either risk-benefit isn't favourable, or therapeutic efficacy isn't substantiated, or the quality of the product isn't as declared. It doesn't mention p-values, it doesn't mention posterior probabilities. So there's nothing in legislation that says how you've got to go about doing this. Really important for me, and Scott mentioned it, and I disagree with the context. I think Karen's going to mention it in your slides, and I disagree with it, is the link between p-values and decisions. And Deborah sits on our National Advisory Committee, I sit on the European Advisory Committee, and there is not this direct link that says 2 p-values less than 0.5, approve, don't have 2 p-values less than 0.5, throw it out. It's nowhere near so simple. It's true, of course, that people summarize the strength of available evidence using p-values, using estimates, using confidence intervals. But it's not so closely linked to the decision. If you come along with a new drug for asthma, for type 2 diabetes, and you've run 2 trials, both of which fail to meet the conventional nominal significance levels, you probably won't get a license, because it will probably be concluded that you don't have the right strength of evidence. Okay? But that could apply to a Bayesian world as well. If you come along with posterior probabilities of 94%, because we'd still have a cutoff. So the debate there is, what cutoff are we using, not what method are we using. So I'd argue that the decision making is rather distinct from the statistical tools. You can criticize the decision making, if you like, because it's subjective. It's about whether a panel of experts, whether a panel of experts are committed, are convinced enough to press the green button rather than the red button. But whether or not you like it being subjective, it's certainly holistic. What do I mean? Really wish I hadn't pictured a ruin. The metaphor falls flat. But the idea of this was to say how the world works at the moment, using a frequentist framework. So these pillars, which are holding up what's left of the rest of the structure. So this is my drug license at the top. And these pillars are external scientific information, exploratory development, and 2 confirmatory trials, just as a standard development program. And that underpins the licensing decision. And the pharmaceutical company, the sponsor, has to do other things as well. But that's usually what underpins the license. in addition to proposals for further study of the drug. And that's the basis for the rest of the arguments I'm going to make once I've drawn my conclusions, which I'd better get on with. So there again is the definition of Bayesian, and here's my interpretation of what's meant by fully Bayesian. It's one continuum of accumulating information until all of the relevant questions have been answered with sufficient confidence. I probably shouldn't use the word confidence. And that the observed data can contribute to several questions. Now what do I mean by that? I mean that phase 2 data, dose response data, would answer a question on dose response and would then be an informative prior for your phase 3 trials, for example. And where am I at the moment? Well, I don't actually call myself a frequentist or a Bayesian. In fact, I rarely call myself a statistician, but that's an aside. And I do think this should be a debate about tools. I think that's the idea of the discussion. Which tools should we be using? Not actually about standards. I think that's a separate debate. Entirely agree with the idea that all of us, including regulators, are really Bayesians. Because when Deborah walks into the CHM, or when I walk into the CHMP, you remember everything that's gone before, and that's your starting point, and that's classically Bayesian, of course it is. I won't go into detail there. Really strongly support the idea that when you hear regulators don't accept Bayesian methods, just check who's been asked. So, breaking this down, this House believes pharmaceutical and regulation fully Bayesian. Pharmaceutical development, absolutely in favour, particularly all the exploratory phases, all the decision making, makes sense, no problem, don't want to repeat what's been said. In fact, it's actually quite painful now to see a 60 patient dose response trial with only 2 doses, with only 20 patients on each, and the basis for inference being a hypothesis test. It's crazy, but it still happens. In fact, it's perhaps slightly contrary to what Scott's saying, it's probably still what we see the most of. But for regulation, I'm against. What I think, I've put this down as the big benefits of frequentist thinking. In fact, that's perhaps not strictly accurate. It's perhaps that the Bayesian doesn't quite fit. We work, the way we think as regulators is this LearnConfirm framework. So the sponsor goes away and they learn everything they need to know about their drug in order to enter this stage of confirmatory development. And then you set the clock to 0. Then you start again and you prove what you set out to prove. You prove your hypothesis. So famously described in the literature as the LearnConfirm Framework, it's one that it's the model we follow, if you like. And that is the antithesis of the Bayesian approach. And a couple of points which are really key to me in terms of the benefits of what we do at the moment. Firstly is the independent replication of evidence in phase 3, at least when that's possible. So you don't only get to a point, set the clock to 0 and prove something. Along with all good science, you then replicate, at least in a reasonably close way. So you've got these pillars, not just the exploratory development, but 2 independent replications that you actually, you know what your drug's doing, you've quantified what your drug's doing, the drug works. And I have a fundamental problem that would need to be addressed if we moved more in the direction of a fully Bayesian world for confirmatory trials, which is the use of informative priors. Because experience tells us that we don't know what we think we know when we start confirmatory development programs. We're all familiar with the phenomenon that if you do a phase 2 trial, in fact you do a bunch of phase 2 trials, and you use the estimate of the effect from that phase 2 trial to size your phase 3 trial, it will on average be an overestimate. And that's because we pick the best of our phase 2 trials to pursue, and we kill the rest of them. And on average, we're overestimating what's likely to happen. It's also a different experimental situation in phase 3. You hear it described as being more real world or more variable, one of those. We also know that there's a bunch of products which are highly successful in phase 2, which completely fail in phase 3. And the success rate for medicine's development, industry know this better than I do, but out of every 10 products that go into man, I think one makes it to market, broadly speaking. And there's a number which actually fail during phase 3. 50% of those failures are because the drug fails to beat placebo. So if you've used your early phase data to construct your informative prior for your confirmatory trial, how reliable is that going to be? Of course, we can play around with the prior, we can increase the uncertainty, we can shift the location, we can do whatever we want. But you might find that to satisfy people for this treatment effect in a confirmatory trial, you have to shift the location all the way to 0. So really what I'm saying is, what do we end up with under the Bayesian framework? This is my picture of the Bayesian framework as described by Roman pillars. But what do we end up with? In my fully Bayesian world of gradually accumulating information, do we end up just with one pillar? Because actually, we've got our background science and we just add a little bit of dose response and then we just add a little bit for confirmatory because the same data is contributing to all these different questions. And so do we just end up with this? Or do we actually end up with the whole huge all singing, all dancing pillar which genuinely does answer all the questions to the same standards that we have at the moment without losing any information that we get at the moment, but just summarizes them using different statistics? There's a bunch of interesting questions about how this would actually happen in practice. I think the only things I want to pull out, okay, maybe we've got 30 seconds. How to agree the priors, who would do it? Sponsors or regulators or agreement between the two? And there was a wonderful phrase in that other session. James, what was the phrase about skepticism, skeptics? It was in one of the responses to your paper, I think. Fitted regulators perfectly. Who regulators, of course, think they have the correct degree of skepticism. But if you get your regulators to specify your priors, they'll be rather more conservative in the industry. And by the way, quite rightly, because I read clinical trial reports every day, and my reading of the inferences that are drawn are not supported by the data that are in them. And if that same rose-tinted spectacles is applied to how you set your priors, there's going to be a problem. We have certain principles that are important, and I want to stress the word principle because whether it's a p-value or whether it's called type 1 error, the principle of controlling for false positives, however you do it, whether you call it frequentist tool or not, but the principle of controlling for false positives is important, and we'd have to find analogous solutions for those. In fact, intelligent people probably already know what they are. In all of this, we mustn't, of course, be driven by bad practice. There's bad frequentist practice, I'm sure there's bad Bayesian practice, and that shouldn't be the driving force. Conclusions. I'm a bit more conservative, perhaps. I've said we're halfway there, because I think this does happen within sponsor pharmaceutical companies who are rational and using the best available tools to make their internal decisions and conduct their exploratory development programs, learning, modelling, making decisions and planning future trials. I think is nice in this framework. As a tool for reporting and decision making in confirmatory trials, if you don't lose the standards, if you don't lose any information, if you can recruit the same number of patients and take the same measurements, maybe it doesn't matter if you call something a posterior probability and set a level for that, or you call it a p-value and a confidence interval. Maybe it doesn't matter at the end of the day. And for difficult questions, orphan indications, subgroup safety, I think there's certainly room to expand in that direction. But fully Bayesian for regulation, I remain against the motion, and with a particular stress on the importance of confirmation and the importance of replication, which I think are against the Bayesian philosophy.
Okay, thank you so much. It's a pleasure to be here today. As was mentioned, my name is Karen Price, and I work with Eli Lilly and Company. So the perspective that I'm going to give today is as an industry statistician I also wanted to mention that I currently lead the Bayesian expert team at Eli Lilly and Company. So I think you probably know which side I'm going to land on before I get to that slide. But one point that I wanted to make is that industry has invested heavily in Bayesian methods. So my full-time job is to help develop Bayesian methods, clinical trials, analyze data using Bayesian approaches. There are 2 people that are in my group. So we have 3 full-time statisticians, including 1 computational statistician. And then additionally, we have another group, which is clinical trial optimization team, which relies heavily on Bayesian methods. So industry is taking note and sees the value in the use of Bayesian methods, and therefore is really putting their money where their mouth is and investing money and resources to further develop. So with that sort of backdrop, am I for or against the motion? Of course, I'm for the motion. I think that one point I wanted to make is one thing we may want to do as part of the debate is exactly what the motion means. So when I hear the motion, what I was thinking is that fully Bayesian, meaning that regardless of the phase of development, a Bayesian solution would be allowed and acceptable. I really feel that there is a lot of acceptance for Bayesian methods in early phase, sometimes in post-marketing type of situations, but I think that statisticians tend to turn off their Bayesian machinery when it comes to confirmatory studies. So most of what I'm going to talk about today are opportunities for using Bayesian methods in confirmatory studies, things that we are doing currently at Lilly. Some of them have been touched on already, but that's going to be a little more about where I'm going to be focused. And with that in mind, I fully believe that our full statistical toolboxes should be brought to bear at all phases. So again, we don't need to be turning off our Bayesian machinery because we're about to undergo a confirmatory study. That Bayesian and frequentist methods bring unique aspects to our understanding of knowledge of data, and all of this with the mindset that at the end of the day, the patient is waiting for important drugs that are safe and effective. And so if we ignore what Bayesian methods can bring to the table, we are potentially slowing safe and effective drugs from getting to market. So as was requested in the motion, what are some of the strengths and weaknesses? So I think from a strengths perspective, there are a number of them. I just mentioned a few here, that drug development is a process of learning, so incorporating that knowledge over time makes a lot of sense. Generally, Bayesian methods answer the questions that we care about directly. There is a probabilistic interpretation, and so that really helps with decision making, and I'll touch on that in a later example. There are opportunities to formally borrow that information. In terms of weaknesses, a couple of things. Obviously, as has been discussed, is that the specification of priors is required. However, I also see that can be a strength because it does allow for more transparent discussion, whether that's within a company making decisions and sitting around a table with various clinicians as well as statisticians at the table to indicate what their current beliefs are. That facilitates discussion, but also with regulators and payers, thinking about those priors, I think is very important. Another weakness I mentioned is simulation, and I almost put this as strength because, to be clear, I think that every clinical study should be simulated before it's run. So simulation is a very valuable tool, but I just mean that, as was mentioned, there is not closed form, and people worry about ensuring type 1 error via simulations. The type 1 error is protected via simulations, so it is a cause for concern. And then, as well as was touched on by Dr. Berry, the formal incorporation of information can cause bias. So in my next couple of slides, I'm just going to talk at a very high level about opportunities in confirmatory studies. Some of these have already been discussed. But obviously, adaptive studies, I give an example of sample size re-estimation here, but there's a number of them. The phase 2-3 seamless, here I am referring to the study that Dr. Berry mentioned. Interim analyses, the timing and conduct of them. In non-inferiority studies, and I will give an example of what I mean by this in a couple of slides. Across the board with evaluating signals, safety signals, detecting and evaluating, as well as assessing dose-related adverse events. And then there are some operational issues around evaluating site performance or predicting enrollment rate or predicting events if it's a landmark-driven study, for example, predicting when that landmark event will occur very much lends itself to a Bayesian solution. And then I have this last bucket of decision-making, And I think this has been touched on quite a bit by the other speakers. But in my next example, I will show you how we're using that at Lilly. And then I just did want a slide to say I certainly, the Bayesian methods are relevant across the development phases, as is listed here. Okay, so the example that I'm going to talk about today is just one example of how we're using Bayesian methods at Lilly, how we're using them throughout drug development and also to inform phase 3 studies and design phase 3 studies. So this is just one example. When we want to assess the competitive landscape, we may do a literature search to obtain papers that are available, and we would have a series of studies. So ignoring this feature study, just on the left-hand side, we'd have study 1, which say there was an active comparator A against placebo. Maybe there was an active comparator B. In study 2, you see it's placebo versus active comparator C. And you'd have additional studies with maybe placebo active comparator C and additional active comparators as well. And what this picture is trying to describe is, suppose that we're imagining a future head-to-head study with active comparator A and versus C. So what we would do at Lilly is we would use this information to inform what that study should look like. And I had to take away a lot of the labels in order to show this slide, but this is something that we look at Lilly on a regular basis. So these are posterior distributions using a Bayesian network meta-analysis of the framework that I showed before. And so you can see a lot of things here. Placebo is on the right, and then supposing that compound A and C is what we're interested in, you get posterior distributions for these compounds. So immediately you have information on the estimate, on the variability. We can also obtain posterior distributions of the difference of these compounds and calculate posterior probabilities that they exceed certain clinically meaningful margins. This example is a change in A1C, so you can see this is percent A1C on this axis. And as we're making a decision about moving a compound further in development, we would use this to account for where we are in the competitive landscape, and then also what is the variability associated with our estimates. One thing that I wanted to mention, since was mentioned earlier, we would also then use the posterior samples coming from this analysis to simulate the feature studies. And we will take this information to our decision makers. So these have been presented to high levels of management, saying here's our posterior distributions, here's our posterior probabilities, and our predictive probabilities of success. And important decisions about phase 3 studies have been made using these types of analyses. And it's a statistician who's at the table describing the approach, and I think is one way that we're seeing a statistician take more leadership in making decisions within industry. And this is just, if you can't see, it just says I figure there's a 40% chance of showers and a 10% chance we know what we're talking about. This was in a BMJ article, and I just think I use this sometimes when talking particularly with clinicians to say presentations such as this are very important, because it doesn't just matter what our estimate is, but we really need to understand how sure we are of where we are. This next example, then, building on the evidence synthesis theme, is a proposal for how Bayesian methods could be used fully and in a phase 3 study. This would be in a non-inferiority study. We have had some discussions with some people at the FDA about this, and they seem to be favorably disposed to this at this time. But the idea here is that you've got, this is an example where suppose you have 4, 5, 6 studies for placebo versus compound A, and then as it comes time to do a study for compound B, it's ethically you cannot compare against placebo. Well, in the non-inferiority guidance, what is generally done is there will be this evidence synthesis of placebo versus compound A. A margin will be defined, and then the second study comparing compound A and B will be conducted, and we will look to see if the margin was met. What this is doing then is it's, rather than worrying about the margin, it's defining this evidence synthesis Bayesian model to allow direct comparison with placebo. So you can see here, we just we implemented this just using some data that was available in the literature, just to demonstrate that with this kind of approach, you can directly obtain a posterior probability that compound B is better than placebo. And if you cared about a margin in this example of 1.5%, you would obtain that probability as well. So that's a proposal that could be done for any non-inferiority study as the primary analysis. Now I got these two out of order. So covering the hurdles, some of the hurdles that I see as it being an industry is the lack of experience by statistician, as well as lack of knowledge and comfort by clinicians, decision makers that we're working with. So certainly there's a perception by statistician that I don't want to take this because they won't understand. So I'm not going to worry about that. There are certainly concerns that regulatory will not accept. I think many of those are being alleviated, but at the same time, with that concern, decision makers in a pharmaceutical company may not want to use a Bayesian approach sometimes. For a statistician, a lot of times there's too many priorities, and so we get that a lot, that statisticians are just trying to do what they have to do to get the basic stuff done, if you will, and so trying to think about learning about Bayesian methods and developing a Bayesian trial is almost too much. So that's where the setup like we have at Lilly, I think, is very effective in overcoming this, in that we can come in then and fill that resource gap. So those of us who are on the Bayesian team can help fill that resource gap so that all statisticians aren't having to learn about all of these things. And certainly there are computational challenges, although I think that's improving. So how to overcome a number of things, I think, certainly education and communication. And then I wanted to go back to this previous slide and clarify a point. So I think that there are a couple of things. One is when we submit to regulators, again, there's this perception that a key success criteria for submissions is 2 phase 3 studies at P less than 0.5. However, and I didn't mean to indicate that that's somehow you get a green automatically. But what I do think is that secondary analyses for the primary endpoint are important. The secondary endpoints add to our knowledge of the data. Those can be analyzed in a Bayesian way, and that can be provided to regulators. Because I don't think it is so clear-cut that you have 2 phase 3 studies and you're done. We really have to understand the data, and we have to be able to communicate to regulators our interpretation of the data, and a Bayesian interpretation would provide valuable insights for all of these points here. And so I think as statisticians, a step in the right direction would be to continue to provide these analyses in submissions and to regulators. So one solution. Another solution that I wanted to just mention is that, again, in line with the education and communication, I currently chair the DIA Bayesian Scientific Working Group, and we are a group of representatives from regulatory, academia, and industry, engaging in scientific discussion and collaboration, with the goals to facilitate the appropriate use of the Bayesian approach, as well as contribute to the progress of Bayesian methodology. And our mission is to create a scientific forum for discussing and developing new tools, providing education on and promoting dissemination of methods and best practices for Bayesian methods, and I think really, really importantly, engaging in dialogue with industry leaders, the scientific community, and regulators. So why are we doing this? Well, we feel that Bayesian methods provide a framework for leveraging prior information as well as data from diverse sources. And only by bringing all of these groups together will we be able to overcome whatever hurdles remain, whether perceived or really hurdles that we're encountering. And that a coordinated effort is needed. One of the nice things about this group that as we've formed and made progress is that we're able to proactively engage in scientific discussion to know what guidances are coming and how can we engage in that discussion early on. I mentioned some hurdles earlier, and one of the things that this group has done early on is conduct a survey that was distributed to a number of pharmaceutical companies and academic centers, or research centers. And at a high level, the main survey responses were that the main hurdles to applying the Bayesian approach was insufficient knowledge, lack of clarity in terms of the regulatory position or guidance, lack of good Bayesian examples or precedents, as well as company internal difficulties. So I think that's the perception that the clinicians or decision makers within the company would not be supportive. In terms of preferred venues for Bayesian education, live face-to-face is preferred. So courses such as a conference like this over less interactive trainings is preferred. And you can see the Bayesian topics of interest there. But I wanted to mention this to say that the surveys seem to back up a lot of what we're feeling in terms of the perceived hurdles, and there are a number of things that are underway to overcome these. And so in summary, the Bayesian approach provides a framework that allows us to leverage information and enhance decision making. We can optimize drug development at a variety of levels within a pharmaceutical company. Bayesian methods are directly answering the questions that we care about and allows for flexibility, can easily predict future outcomes, and have a straightforward interpretation. And I believe that the Bayesian approach contributes to our overall assessment and understanding of data, and therefore should be considered at all phases. And just to close with a couple of quotes, you may know the famous baseball player Yogi Berra, it's tough to make predictions, especially about the future. And I think that Bayesian methods can help. And then lastly, from Rubin, the applied statistician should be Bayesian in principle and calibrated to the real world in practice, which I think is what we should be doing.